start. All right. Put up the floor for questions related to lab stuff. First, anything we did today you want to get clarification on, or?
Can I ask, as far as like, I know that when I was looking at the heart, the apex is in the bottom as I'm looking at it, the right side, but it's really the left, correct?
In you and I, it's tilted slightly to the left, correct?
Okay. So then, like, I did make a couple wrong cuts. I was just trying to look at them after I got home.
When you say wrong cuts, you probably, what you mean by that is you cut into the interventricular septum.
Yeah, I opened up. Yeah.
Yeah,
exactly.
and that's a common thing to happen the first time you've done it.
Okay.
And when I asked you to cut through one of the auricles, I didn't know half you're going to pick the right, half you're going to pick the left.
Well, and wasn't there more of one auricle than the other?
Huh?
Typically, I thought the top right one, when I was looking at the other girls' hearts, ended up being like.
No, they look pretty identical.
I just thought one was bigger than the other, I guess.
No.
Have to be there more than the other. Okay.
No, you really, for most people, you can't, unless you have a lot of the extension blood vessels on the top intact, it's really hard to decipher right or left half when you look at it. So that's why you've got to cut into it, and then you know.
Right.
By cutting into it, half of you will cut into the left side, half of you cut into the right side.
Right.
If you cut into the left side or the right side, which you did,
right,
you're going to probably likely cut into the interventricular septum. I can't tell half of you to go halfway down and half of you to go all the way down because I don't know which half you have.
I gotcha.
So this is bound to happen. You just need to understand that when you do cut into the interventricular septum, that's what you've done, and that's why you've done it. There's no big deal.
Right. Well, no, I did feel like I just wanted to make sure I was yeah, I'll ask more specific questions later, I guess. Sorry.
Well, no, I'm happy to answer questions. I just don't want you to feel bad having cut into the septum.
Well, I just don't think that it's probably hard for you to remember when you go along and look at each person's heart, like where exactly they might have messed up or where they cut. I know my first one, I cut way through too deep.
Right. And that's because you were on the right half. And when I told you to cut all the way down, you followed directions. And so it was not your fault so much as, but I didn't know what side you were going to have.
Okay. Well, okay. Oh, what I did want to ask is, so like when I have my husband and his friends and stuff donate these, is there a way that I can tell them to cut the heart out so that you can have that pericardium?
Yeah.
So can we talk about that?
Yeah. Just briefly, all you have to do is tell them to not cut too close to the top of the heart. In other words, take some of the blood vessels, maybe an inch or so above the heart itself, and try to cut through those. And by doing so, the sac will sort of remain intact, because when you cut low, you're cutting where the sac attaches to the blood vessels. And so it's bound to slide off.
Strong that was.
Yeah, absolutely.
I mean,
yeah, it is. It's amazing. That's why I wish I could have had one intact to show you, but you got a sense of it.
Okay.
Yeah. It is amazingly strong, and it's amazingly see-through in that sense too.
Right, yeah, because you pulled it apart. I couldn't even I'm sure I could have ripped it if I tried, but it was very strong.
It's tough. It's fibrous. They call it a fibrous pericardium for a reason. Yeah. Yeah. Okay, other questions on the dissection? Okay. Any about stuff regarding lecture? I'm sure there's going to be questions on that, whether it's on the blood chapter maybe, or respiratory. You tell me kind of where you are, and we can talk about cardiovascular too. That's fine.
Can I just ask one more question real quick?
Sure.
My grandma, she had scarlet fever when she was like 7, and it damaged her heart. So she was like one of the first people to have this like experimental surgery where she survived a partially plastic heart. They told her she'd never go through labor. She had 3 children. She'd never watch her grandchildren, all this stuff. And I just wondered, like, sort of what part of the heart, this is like, I'm talking 80s. She died when I was 7, so that's 86. But she literally survived one of these first kinds of surgeries. And she had partially plastic heart. What part of that heart would have that been?
I don't know. Part plastic heart. I've never heard of that.
Well, I mean, like,
Uh,
fever, what would that do to your heart as far as damaging it if your fever was super high?
well, I'm trying to picture what you're describing, what she would have had done. I mean,
I almost think it was like
um,
the wall between both sides.
I just don't know. You'd have to find some more information about it. They
Right?
did back in the 80s at the University of Utah. They did some pioneering work on the artificial heart, where they actually implanted an artificial. It was called a Jarvis 4 at that time, named after the physician that came up with this. He was a cardiologist.
Uh,
And again, you guys are too young to remember, but there was a dentist, a retired dentist by the name of Barney Clark. He was the first one to receive the Jarvis 7 or whatever it was called. And he lived for, I think, a week. He was in the hospital. He did not go home. And then there were several other individuals who received that same heart. The technology and the engineering at that time, and even today to some extent, it's so hard to create an organ or a system like that, an artificial system that can be as efficient as our heart is, because there's so many variables you have to contend with. It's a real engineering challenge.
yeah, I mean,
But in the last year or two, I heard on the news that they're revisiting going back to trying to re-engineer an artificial heart. So it kind of was off the table, so to speak, in terms of research, I think, for a number of years, but it's now being investigated again. But it's hard. It's very difficult to come up with a substitute.
imagine. Because I mean, that tissue forms in relation to the way your body has like
Yeah,
pumped cells.
yeah, exactly. How do you devise an artificial system that
Our-
pumps appropriately to make up for the fact that you're running a race, or you're sitting at home reading a book, or you're sleeping? I mean, it's very complex.
And it's so unique in each one, even though it's all the same still.
Yeah,
You
exactly.
know?
I have a question. So I know we can take like components from like pig hearts or something
Yeah.
like that. Is there a reason why we can't just take like a whole pig heart and do like a heart replacement with that? Is it just because they're too different, or?
I think it boils down to rejection issues. Yeah. I
Yeah,
think
because
so.
don't you have to go through a lot of like blood typing to see if they're like an eligible donor and things like that?
For some types of transplants, absolutely, like bone marrow, you have to have a match.
Mhm.
I don't know enough about organ transplants and the difficulty in terms of rejection. I'm sure some organs are easier to transplant than others. For example, a liver is not a big, well, I shouldn't say it's not a big deal. It is a big deal, but I've heard of people who've had liver transplants, and what's cool about the liver is that it's this large visceral organ. It's like the biggest organ of your body. What they can do is take a small piece of it from, let's say, your sister, who's a good match, we'll say genetically. And your body will take that small piece of liver and regenerate the whole liver again from that. Isn't that crazy?
Mhm.
Yeah.
And it's probably hard to find, well, doctors and people willing to be like, oh yeah, I just replaced my heart with a pig heart. Like, we'll see if I die.
Yeah. If you go back in history and study this, I don't ever remember hearing about trying a totally different organism. But I think there's some
Kidneys.
rejection issues, some immunological issues. It could be that pigs just, because they're different species, there's a difference in terms of the immune response that they would have versus we would have. I think that's the major issue.
Going out and seeing these
But what they're
people.
trying to do with biotechnology is to create, you've heard of these 3D printers, right? where you can make about anything you want with that with these crazy printers now, right? To be able to engineer using live tissues, tissue cultures, recreate an organ. I mean, they've already done ears that way.
Because isn't it if you make like a frame for it, like a type of like you can put stem cells or something on it and it'll
yeah, exactly. That's the same idea. Now, of course, an ear is pretty simplistic in the sense that it's cartilage and epithelial tissue, right? But now you talk about a heart, you all know we've got muscle, we've got nervous tissue, we've got connective tissues. I mean, it's much more complicated.
Well, so how do we, I mean, I think it was Paige actually, we were talking earlier when we were dissecting the heart, was about like reattaching like arteries, veins, capillary, those kinds of things. Like, how do they figure out like which ones will regrow or which ones you have to reattach?
Yeah.
I mean, I can't even they're so tiny. I mean, to have to follow around the whole tube to like reattach it just sounds insane.
Well, it's probably not as bad as you think, because if you have a new heart put in, and that's not uncommon, right? People are walking around today with hearts from other people. When you start studying the basic anatomy of the heart, look at the blood vessels, there's the aorta that leads from the heart. There's the superior vena cava coming in. If you can connect the major blood vessels, then all the other blood vessels within the heart, like the coronary arteries and so forth, they're already in the heart. So it's like you're connecting the major inputs and output blood vessels. They're not replacing all of the small intricate ones.
And then go ahead.
I'm sorry for you. No, this is just interesting. My friend was telling me about this book where, they are doing studies that there is so much nervous tissue in your heart that they're saying that it can hold they believe that it can hold some like memories or some imprints of like who you are. And so she was telling me about this whole book, and it was just a compilation. It was like a huge book of people who had heart transplants, and about how all of a sudden they had, liked foods that they never liked before, or they felt like they had been to Chicago, but they had never been to Chicago, and things like that. So I thought that was kind of cool. I'd like to track that down.
Wow, that's crazy. I've never ever heard of that. Wow, that would be amazing.
Yeah, I'll have to try and see. I'll if I find it, I'll tell you next class.
Yeah. Wow, that's borderline sci-fi. I don't know, but
I mean, don't you think it's like a border between where you have to like incorporate like your instinct of what's going on and like the science part of it? Do you know what I mean? Like, I think like one of my favorite things about you was, I think earlier I said something about mutations. You're like, yes, because I'm like, they're not always bad. That's like the variation is the basis of evolution.
Right.
So I mean, like, how can we even like assume we understand these kinds of things, Incredible.
Well, we know a lot in terms of medicine is because we've
but I think we know things because we don't know them, quote, scientifically, when we kind of almost instinctually could like just look beyond them. Like, I almost feel like our subconscious has like a connection to like if you put like conscious thought to, involuntary things. Like, my mom has MS, so a lot of times they tell me to keep making her do the physical part so that it's in her brain, even though she can't do it with her foot. And just that sort of thing just was so interesting to me. So you're not losing the whole thing, just half.
You know what? What you should do is go online, look up PBS American Masters, and watch the segment on what's his first name? His last name is Sachs. It was just on TV last week. I watched it. It was fascinating. Okay. He's a physician.
Like PBS.
Pardon?
PBS, my favorite channel.
Yeah, PBS. It's American Masters is the series.
Okay.
And what is his first name? Edward Sachs? No, it's not Edward. Anyway, he SACKS, I think. Okay. Anyway, this talks about his contributions. again, we're getting off topic here a little bit, but he had patients in psych wards and in other places that most physicians would not go and visit. Anyway, it gets into some of what you were talking about. It's fascinating. I think you'll really enjoy that 2-hour show.
Okay,
Really fascinating.
thank you.
Okay, so back to A&P, which
Sorry.
no, this is all good stuff. It's fun to talk about this. Topics you want to review, you want to go over. I'm going to give everybody an opportunity to ask questions or whatever.
So with like the exam, so the couple like past quizzes, like you told us to like focus on like certain sections. So we also do that for the exam, or should we just know like the whole chapter?
What's the easiest way to respond? Just know everything. No, let's talk about that. So we've got, let's see, what chapters?
I think it's like 8, 9, 14, and 19.
19. Okay, yeah. So 8 is on joints. So let's take a look at joints first. We'll just kind of peruse through those chapters and see what we can come up with. A large part of chapter 8, at least the last third of the chapter, is devoted to specific types of synovial joints. And so that is not something you really need to worry about. And the same, of course, applies to the muscular system. So that must be John.
John sounds like a little.
John Chipmunk. You need to reboot, John.
John.
You're chipmunking again.
He's such a hard worker.
Poor guy. We just have to razz him as much as we can, right? He takes it well. All right. So let's see. I'm going to share my screen. Why do I have integumentary system? That's not what I want. What the heck? Joints. Is John back yet or not? No. Here he comes. We should all just move our mouths and just fool with them. Make them think we're talking. We're not. Oh, aren't I mean?
So all of this is coming from the book, correct?
Yes, that's right. And I've got the PowerPoint for chapter 8 loaded, so you should see that. And so what you should be familiar with, of course, is the fact that we talk about joints. You know, we classify joints basically in 2 ways. We can talk about the degree of movement within the joint or lack thereof, and that's what this talks about, right? I'm not going to go through all of these, but this is the functionality classification approach, if you will. And then what's the other approach? Well, that is what kind of tissue is making that joint up. It's an anatomical, so it's like a functional approach and then a structural approach. And I think you should be familiar, you know, with these terms: synarthrotic, amphiarthrotic, diarthrotic. And then we, of course, went through the 3 major types of structural joints, which you see here in blue. And we gave examples in the body of where you can find those. And that's really what the PowerPoint walks us through, right? Now, what do I need to know? Well, you kind of need to know all of this stuff. I know it's a lot, but it's palatable. You can cut it up into bite-sized pieces, right?
So from what slide to what slide? Because I copied these.
1 to 51.
So know these slides.
yeah, these again talk about the functional and structural classification schematics. We did spend time talking about the knee joint as it related to it being a synovial joint, if you recall. Now we did this in lab, I know, but we also talked a bit about it in the Zoom. So I think for this purpose, you should be familiar with the knee joint. Now you don't have to remember all of these different ligaments that I pointed out here in green, so don't worry about that. You don't need to know the name of this muscle. All this is designed to do is to give you an idea that even as we go, out from the internal structures like you see here, there are additional ligaments that wrap around the knee and provide further structural support for it, because it's a pretty exposed joint. It's a pretty complex joint, and this is the same basic thing we find in the shoulder or the hip. There are additional ligaments that help reinforce the capsule. And then
I'm sorry.
Go ahead.
The slide before this one had those the one, well, no, not that one. The one it was a fake one. Yeah. Like the anterior cruciate ligament and stuff, those coming out. Are those coming out like from behind and like up in the femur?
Yes.
Okay.
Yes, they insert kind of on the posterior side of these condyles, right? Just exactly like, I think the sketch does a decent job kind of illustrating that here, yeah. Now the posterior ligament, of course, as the name implies, we can see it anteriorly if we flex the knee, and this of course illustrates a flexion situation, right? But these ligaments, they insert into the bone, into the periosteum of the bone. Yeah. So we described specific types of synovial joints. That's really kind of what this slide summarizes, right? Ball and socket, condylar, hinge, plane, pivot, saddle. Those are the 6. And then finally, we wrapped up the chapter talking about specific types of movements. Abduction, adduction, flexion, extension, right? Supination, pronation. So be familiar with those that I presented here. Again, a lot of these also showed up in lab, didn't they? Yeah, so there are some topics, John, that we're going to be learning in lecture that we also apply to lab. So there's overlap in these examples. But for the test next week, this is lecture stuff. So it's also helping you later on the following week. Did we do joints already in lab? I'm trying to remember. Did we do a practical on joints?
Yeah, it was on our last practice.
Oh, okay. Yeah. So I was going to say, well, you're going to see it again, but I guess you won't in lab. So there's chapter 8. Just to
So,
be clear, go ahead.
This chapter is kind of overlap with the lab practical, so it will be on the test for next week. Is it correct?
Chapter 8 is on the 3rd lecture exam, but
Yeah.
we already tested it in lab on the 2nd lab practical.
Okay, so but next week for the exam is still on the test.
Yes.
Yeah. Okay.
So make sure you go to the syllabus.
Yeah.
Yeah.
I know that, but like because we already have this chapter, it have in the lab practical exam, so it kind of overlap and have to study again.
Yes, that's true. And I don't like to have you have to do that. It just so happened this is the way it worked out.
I think because the computer and coronavirus and stuff. But yeah, if you want to talk about that, I will talk about that because I need help with that too.
Okay, thank you, Freya.
Okay.
Just make sure that you're focusing on chapters 8, 9, 14, and 19.
Yeah.
Yeah. Okay, so let's go to chapter 9, unless there are any questions on 8. And 9 was the muscular system. Okay, you all see that? Muscular system? No? Okay.
Yeah.
Oh, you do?
Yes, I did.
Sadie was saying no, so I didn't know. How about now?
Yeah, now we do.
Okay. So again, what this chapter does is it begins talking about different levels of organization within skeletal muscle. Because you can't understand the contraction and relaxation processes. You can't understand the concepts of contraction at the myofilament level unless you understand some of this basic anatomy. And so this is that summary slide that we've talked about during review, I know. And it takes into account, 4, 5, 6 slides all in one, right? So it is important to understand what a sarcomere is made up of, to understand the overlapping and lack thereof, perhaps, of some of these thin and thick filaments, right? And then, of course, this leads us to talking about how these interact with one another during the time of contraction and relaxation. And we know that this process originates way back in the neuromuscular junction. So we talked about that. Right? And then we discussed the sliding filament model or theory. And there was a couple of videos, I think, here that kind of showed you some animations of that interaction between the myosin and the actin. And in your book, there's a series of tables, or a table, I guess, that summarizes the major events. So this certainly would be important to know. If I were to ask you on an essay question, describe describe the way in which actin and myosin interact with one another during contraction. I mean, that's a big question. Or I might say, describe the steps involved in muscle fiber relaxation. So you'd have to go all the way back to the neuromuscular junction, wouldn't you, and talk about how the acetylcholinesterase would degrade the acetylcholine, and that would then prevent any additional muscle impulses from being generated. And that would cause the calcium to be then actively transported back into the SR. And if you remove the calcium from the troponin, then the tropomyosin moves and blocks the binding sites on the actin filament.
Tropomyosin that moves, or does the band twist more?
When you remove actin from troponin,
Right.
the tropomyosin slides over and blocks the binding sites.
That's what moves, though, the tropomyosin?
Yeah. Yep. Yep. Because if you go back to the diagram here,
Grabs it, right, and pulls it along. I'm sorry.
where is the, oh yeah. So here we see in the actin, both the troponin in the 3 protein subunits here, see? And then here's the tropomyosin in purple. And they're blocking the binding sites, aren't they, on the actin? Are you seeing the diagram? Yeah, okay. So if there's no calcium attached to the troponin, which there's not here, you don't see any calcium here, do you? Doesn't say Ca++, no. So if there's no calcium bound to the troponin, then the other complex protein here, the tropomyosin, is blocking the binding sites. But when we introduce calcium, like we're going to do here, there we go, calcium comes on board. Look what happens to the tropomyosin. Here's the before, here's the after calcium. Now the binding sites are open, exposed. And the cross bridges here, these globular heads of the myosin can come up and grab on, can't they, to the actin at those pink sites, those little circles. So you should understand the sliding filament model. It's a very important tenet of muscle physiology. And then we got into some discussion of creatine phosphate and kind of what it does in terms of providing an initial source of ATP, but it only lasts for a couple seconds. The majority of energy production, occurs in the aerobic cellular respiratory process. That's where the maximum number of ATP get produced per glucose. This we talked about back in chapter 4, if you recall. And I don't want you to rehash all that. We've talked about it, but I hope when you see this diagram here, this doesn't look like, Swahili to you, or Russian, or Greek. It would have back August 24th, maybe, or maybe not. Maybe you remember this from high school, but again, I think to myself every year when I teach this, should I stress the cellular respiration process as much as I do in A&P1? Because most instructors don't do that. They spend 10 minutes on it. I spend an hour and 15 minutes on it or more, because it keeps coming back to you. It keeps coming back again and again. And when you take micro, you got to know this from microbiology anyway. So you're going into nursing, you got to take micro. So I think it's important, obviously. But anyway, and then we got into some discussion of oxygen debt and how that forms and how that has to be repaid. And then we got into benefits of training. We talked about some specific changes that we can see in the muscle tissue of athletes. It's interesting, isn't it? And then we talked about some physiology, some concepts. Now, again, this is somewhat nebulous in the sense that you can't see this happen. I'm describing this to you. This is where we probably should have gotten that little machine out and hooked John up to the machine and stimulated his arm and all that stuff, made him dance around. Because it would make more sense, because you'd see it happen, right? It's like the dissection today. It's one thing to talk about the heart, but it's another thing to get into it and see it. So I really wish that we could have done this, but with COVID, we just can't.
I'm just gonna ask you, do you feel like this has been like more frustrating for you to try to like?
Well, sure. I feel bad for you. That's what I feel bad for. It's hard enough to teach it. Yeah, I mean, in this modality, it's hard. Yeah. But I feel bad for you guys, because I put myself where you are.
Well, and like you're like, but you've been educated like older than I was, and I'm terrible at computers, so I can imagine you've got to spend like triple the amount of time than you used to, and then.
Well, I've had to embrace it because I feel that's my job to do that. Plus, I find it interesting, but I am no computer wizard at all.
Well, it's sort of part of our subject, really, right?
Pardon me?
The corona and like what's going on and our body's response is sort of part of what we're studying anyways,
But, well, it really is. It's too bad we don't have time, or maybe I could take something away and put something in to talk about COVID. Because I mean, it's who we are. It's the history we're living through. It's reality. Take microbiology. You'll hear more about it. And then we got into smooth muscle and cardiac muscle. A little bit of commentary about how those other two types of muscle are somewhat unique, though they share some similarity with skeletal. They're all also very unique. And that is kind of where we wrapped up 9. And then, are there any questions on anything I said in 9? Is this helpful, or am I just wasting our time doing
No,
it?
this is helpful because I'm taking it all down, so I don't have to call you 50, 60 times.
Yeah, so like, the slides basically end at like 9.5 then, and like 9.6 is like skeletal muscle actions, but you don't have that really in there. So like, should we just not have to worry about that then? Like the rest of the chapter just goes through like more of like lab stuff.
Right. So 9.5 is cardiac muscle.
Yeah, it like ends at that one.
Yeah. So at 9.6, you're exactly right, where it starts skeletal muscle actions. That is, from that point to the rest of the chapter is lab, right? So again, let the PowerPoint slides be your guide, right? And if you don't know, ask.
Well, and then also those questions you always put up that, you know, like I think Paige and I, there's 2 pages I know, we're talking about like those, you put the questions up and then the answers are there too. Those seem to be like pretty right on. So is that
Okay, what are we talking about?
not,
Objectives.
it just says like the questions, and then right under it's the answers for the whole chapter. It'll be like you can print out 16 pages and it'll just have like a blank line.
Oh, the study outlines, I think.
Yeah.
Yeah.
I kind of stuck with those, but I think I tried to spread myself too thin. But
Are
those
you
right on.
suggesting you find those helpful?
Yeah, I do. But then like I always just feel like I have so much more. So I'm not like focusing on the right stuff, but I did realize once I focused more on those that was exactly kind of what you followed. So
Well, exactly right. I mean, anyone would tell you that I follow the textbook fairly closely. Now, that's good and bad in some ways. It's good for you because you can follow it clearly. There shouldn't be a whole lot of confusion as to, you know, how I'm presenting this chronologically, right?
Right.
Um,
I just meant like if almost like instead of reading through the questions, if you don't have the answers, even if you just read through the one that has the answers of that study outline, it focuses on the main stuff. And it's almost like you're outlining,
Yeah.
you're highlighting just the more important stuff as opposed to like trying to wade through it.
right.
Does that make sense?
Okay. Let's then go on to respiratory. Oh, before I forget, don't forget this animation video that walks you through a lot of the structure of skeletal muscle, and then of course it talks about the mechanism of contraction and relaxation. You have all looked at this, haven't you?
Mm-hmm.
Yeah. So in respiratory, as all the chapters do, they begin with some discussion of anatomy. Now here, we do have overlap between lecture and lab in the sense that as you're studying this stuff for the lecture exam, you're also studying for the lab practical. So you're killing 2 birds with 1 stone here.
Wait a minute, ain't blood 14? Then we go 8, 9, 14?
Oh yeah, I'm going by the schedule is all. You know what I'm saying? We did respiratory before we did blood.
Oh, okay.
I'll come back to blood.
Okay.
Yeah. So, and these are structures we went through and labeled in lab, I don't know, 2 weeks ago or whenever it was. Right, the larynx. Remember the larynx model? We talked about that. So you're again studying for both the lecture exam and the lab practical when you're doing this.
Shit, that means I gotta read the entire.
And so this delves down into the structures of the respiratory tree. So these are the bronchi, the bronchioles, terminating with the alveoli, right? Here's a cast of the bronchial tree. You see how intricate the passageways are as they come from the alveoli, toward the trachea or vice versa. And here we see the myriad of capillary beds here on the surface of these alveolar clusters.
I don't see that. All I see is,
Yeah, your blackboard.
On your blackboard, sir.
yeah, it's frozen.
Okay, sorry. How about now?
Hold on, it says your yeah.
Yeah, I
Good.
got it.
Okay. So here's those, microscopic alveoli that terminate at these alveolar sacs. And we can see the presence of smooth muscle here that line some of these bronchioles. So if they contract, if this smooth muscle contracts, what happens to the lumen of the bronchiole? Does it get bigger or get smaller?
Get smaller.
Get smaller, right? What'd you say, Freya?
I just said tighten.
Well, the
You know-
lumen is the space through which the air is passing. So if these contract, the lumen gets smaller. If you
Oh,
wanna
yeah,
call that
yeah.
tighten, I guess that's tighten.
Okay, I'm sorry. Yeah, I guess I was looking at the other. Okay.
And so there'd be less airflow, wouldn't there? So somebody who has an asthmatic attack is experiencing diminished airflow in and out of the lungs. They can't breathe, they panic, right? You've heard of these, haven't you? People who have these attacks, they're scary. So you take the vasodilator, the little bronchial dilators, right? They carry them with them, and they have the spray, right, or whatever it is, and it relaxes this smooth muscle so that the lumen gets larger, and it allows for much easier breathing for the asthmatic. That's how those work. A lot of them, anyway. Here, now that we've talked about the heart, this is going to make more sense. Here we have oxygen poor blood coming in that emanated from the pulmonary arteries, right? As it left the right ventricle.
Right. Yeah. Okay.
And it's coming to the lungs. And here it's dumping off a lot of that CO2 and it's pulling in the oxygen. And then it's going to be taken back, right? And where does this eventually go? Which blood vessel will this enter? As it enters the heart, what blood vessel carries this oxygen-rich blood from the lungs? The pulmonary veins, 2 from each lung, right? Yeah. And then we talk about the movement of gases across the respiratory membrane, right? We've described that already. So you know kind of what that involves, I hope. Then we talk about the mechanism of breathing, both inspiration, the active process of inspiration, and the passive process of expiration. And you should understand the role that various nerves, like the phrenic nerve, plays in causing the diaphragm to contract, which creates this subatmospheric pressure in the lungs, and air moves in. This is all described in the Zoom lecture, right? Not that I'm unwilling to talk about it again if you have questions on it, but I'm assuming you've watched this. And the contraction of the diaphragm, is the major respiratory muscle helping in inspiration, but we also can tap into additional intercostal muscles, all of which act to increase and expand the rib cage. Well, if you can increase and expand the rib cage, you create an even lower subatmospheric pressure, and more air can therefore come into the lungs. And like the major steps of contraction and relaxation, here we have the major steps of inspiration. Certainly worth studying these tables. This could be a good essay question. Describe the major events of inspiration. That's an excellent essay question, I think. Do you agree?
Yeah.
Expiration is generally passive. What does that mean?
It like doesn't call upon a lot of like muscles to inspiration calls like on a bunch of muscles, but this one like doesn't
Right?
because
Oh,
of the elastic recoil.
When you say call upon a lot of muscles, you mean to have them contract or relax?
To contract.
Right. Now, in expiration, you do have relaxation of the diaphragm, but as you said, Sadie, the majority of the force behind removing air from the lungs is due to this elastic recoil, it's called. All you got to do is think about a rubber band or this ubiquitous thing we wear every day. It's got elasticized material on it, doesn't it? So let's think of this as when you inspire air, what do you do to the elastic tissue of the lungs? You stretch it, right? And then when you're done stretching it, it's going to recoil back, and that's what allows the air to leave the lungs, basically. It's the elastic recoil of all these little stretched tissues. However, this is assuming tidal expiration. Now, I hope you all know what I mean by that. What if I wanted to tap into my expiratory reserve volume? Now, see, we're starting to link terms and concepts. If I want to exhale more air than I ordinarily would via elastic recoil, what must expiration become?
It becomes active because then it calls
Right.
in your inner intercostal then.
Right, exactly right. It becomes active. That's exactly the right word. Meaning, if we want to tap into our expiratory reserve, we've got to stimulate some muscles to help pull the rib cage down and in as we push more air out. So, as I mentioned, I think in class, if you want to play the trumpet, the trombone, the tuba, if you want to become a professional party balloon blower, you've got to push air out of your lungs. That's what we're talking about here. Yeah. And there are certain muscles that we can tap into, including all of the abdominal muscles. Now, here we go learning about muscles now. Remember the 4 abdominal muscles. They're thin, right? They lie right next to one another. And you see them listed. I should hide the slide and say, name the 4 abdominal muscles. Could you tell me? Well, I think you could come up with most of them. Yeah. And then we talk about this material called surfactant. This is really interesting stuff. It relates to how easily our alveoli reinflate after they deflate from the previous expiration. If we didn't have surfactant to reduce the surface tension of water here, we'd have a hard time breathing, and that's what happens to preemies because the respiratory system is one of the last organ systems to form in a newborn. Should call it it's not a newborn yet, but in a almost full term in a fetus, I guess, a late stage fetus. Here comes my friend. You know what I'm talking about?
Yeah, the train guy.
I'm going to wave to him when he comes by because 12:04 every Wednesday, he comes by. I should really give him the finger is what I should do. He makes it so difficult to lecture. I can't talk over that horn.
And
Okay, so in preemies, oftentimes, they have jaundice issues and other problems, which is not related to respiratory, but sometimes they can't breathe very well. And so they got to be brought back to the hospital or kept at the hospital and put into a little enclosure where they're spraying a fine mist of this artificial surfactant, which helps the baby in terms of being able to breathe. It allows for the reinflation of these little microscopic alveoli that are still developing, because again, if the baby's 6 weeks early or whatever, you know, that could be a potential issue.
And that can cause problems like longer than that. Like aren't preemies more like they have more of a tendency to have asthma and stuff?
I don't know. I wouldn't be surprised if that's indeed true, but I'm just not sure. And then of course we talked about these various respiratory volumes. So this is, you know, this stuff.
So could I ask what is the surfactant mean? Is it the name of the cell in the lung or something?
No, it's the name of a chemical that's produced by special cells here that live on the inside of the alveoli.
Oh, okay. It's
So
the chemical.
it's a chemical, right?
Oh.
Yeah.
Right.
And
Doesn't it move slightly against each other?
say that one more time.
Help them move smoothly against each other?
The alveoli.
Yeah.
Well, no, okay. A couple things here. We're talking about this chemical on the inside of the alveolus. If you're talking about, you know, this scenario here, let's go back to the diagram.
Sorry.
No, that's okay. If we're talking about this, do you remember the video that I played in class today? It showed the alveoli inflating and then deflating. There was a segment of that video that talked about that. If you could watch your lungs as you inhaled and exhaled, you would be sitting here looking at yourself all the time, because it's really amazing. They inflate and they deflate. Now you can't see these are microscopic, but pretend you could see them. So what I'm trying to get at is the comment you made, Freya, about something about lubrication, did you say? So when these alveoli, which are right next to one another, when they inflate and deflate, there is a film here on the surface of these alveoli, so they don't rub against one another as they inflate and deflate. But there's also moisture inside as well. I mean, this is a very wet sort of environment.
Absolutely. Well, yeah.
Yeah.
So, okay, but it's not the same. It's not the surfactant that's on the
No,
outside.
no, the surfactant's on the inside. There'd be like they defined it as a serous fluid, I think.
Oh, okay.
Yeah.
Like a synovial almost.
Well, no, synovial is joints.
Right. I just didn't know.
It's synovial-like in its lubrication property.
Right, I just thought that, yeah, synovial was like talked about, like kind of a serous part.
Well, there are membranes called serous membranes.
Yeah,
Okay.
It's a different animal.
Sorry,
closely related.
Combine a bunch of stuff. Confusing.
And so you can go back in the integumentary system chapter if you want to look it up.
Yeah, right.
I can tell you exactly where.
All right, I'll find it.
Page 159, 160. Okay, and then the chapter in the PowerPoint begins to talk about how breathing is regulated by the brainstem. And that's what I've circled here in purple. The brainstem, which you're going to learn more about if you take ANP2 when we get into the nervous system, is composed of two major parts. The pons, shown here, this kind of oval structure, and then just inferior to that is the medulla or medulla, some people pronounce it, oblongata, or just medulla or medulla. Collectively, these two structures are making up the majority of the brainstem, which consists of nervous tissue that regulate the breathing process. And so the PowerPoint talks a little bit about these two respiratory centers, one in the medulla, one in the pons, and how, again, they communicate with various respiratory organs, or muscles, I should say, to help regulate both the duration and the rhythm and depth of breathing. You know that when you're sleeping or driving to school in the morning or whatever, you're not having to think about breathing, you just do it. It's under the autonomic nervous system. It's automatic. It's all automatic. Now, can you override that consciously? The answer is yes. We can tap into our inspiratory, expiratory reserves, right? We can hyperventilate if we want to do that. I don't think you should, but you could. Go ahead, Freya.
Can I ask you that? So at one point it was like you can increase your, by hyperventilating, you can increase the length of time you could hold your breath.
Yeah.
I'm a swimmer, so that just kind of, I never heard that before. So if you hyperventilate, but then it also contradicted, in my mind, contradicted itself because it just seemed like it was, it lasts shorter, the period of time that.
If you are trying to get that quarter in the deep end of the swimming pool, then you're going to try to hold your breath as long as you can to dive down those 15 feet or whatever to get the quarter.
Like opposite what I like when you swim, like you want to hold your breath as little amount of time as you can, so you're not like depriving yourself of oxygen, so you breathe. But like I guess in sprints it'd be different. So is that what we're talking about? Because that just confuses me.
When you hyperventilate, it has nothing to do with oxygen levels of the blood. It has all to do with CO2 levels of the blood.
Right.
Because it's to a large extent the blood CO2 levels, or more specifically, the blood pH that regulates breathing.
Right. Okay.
So you know from yesterday's review, if you hyperventilate, you all know what that is, right? When you hyperventilate, what are you doing to the blood CO2 levels in the blood?
Go up.
Did you say go up?
It rise.
No. John, I would like you to hyperventilate. That means
Oh, wait.
hyperventilate means to take breaths in and out very quickly, right?
Yeah.
When you do that, you're blowing excess CO2 out.
Oh, and I started to get like a little black vision. Right?
If your blood CO2 levels drop because you're breathing more frequently and more deeply, what happens to the blood pH?
Goes up.
Right. Right. Why?
Because you're taking in more oxygen.
It has nothing to do with oxygen.
Taking in more carbon.
You're not dropping.
More carbon dioxide out.
No, if you hyperventilate, your CO2 levels are dropping.
You're not dropping hydrogen.
Hydrogen ion drop down too.
Right.
And like the pH level go up.
That's right.
See, I wish I could see all your beautiful faces right now because you're all so smart. Go my way. That was awesome.
That is the only one I know.
No, don't say that.
The reason that she knew that is because she remembered the equation on the whiteboard yesterday, right, Maeve?
Yeah.
See, I can read minds.
Yeah, I wrote down the equation on the quiz today too.
Yeah, you should. You should. You should do that. That's how you get 10s on your quizzes. You got to know this stuff. So when you hyperventilate, let's think about this. It's very logical. When you hyperventilate, you drive the blood CO2 levels down. You're blowing off more CO2 than you ordinarily would.
You're trying to get more oxygen in?
Well, that's probably an indirect reason why people hyperventilate. That's true. But physiologically, what it's doing is it's not elevating the blood oxygen levels. I mean, it is, but that doesn't impact the breathing so much. It's the drop in CO2 levels and the rise in blood pH is what happens. So if you hyperventilate, take a super deep breath and then dive for that quarter, you can hold your breath longer because it takes longer for those receptors to become stimulated to tell the brain, to tell the muscles, I need to inhale.
Okay. Because we used to have to do these breathing exercises to like sprinters or whatever.
Yeah.
Like I'm distant. So like if you're doing anything in distance, you really don't want to deprive yourself of oxygen as you're, swimming, running, whatever. You want to do it at an even level, right? So that you're not wearing yourself out.
Right. Yeah. I mean, I don't know I'm not versed
Well,
on.
no, I mean, I just thought that was sort of like instinctual in my mind anyways. I'm just saying, like, if I'm not holding my breath as I'm like running or like doing whatever, then I'm not depriving myself. So therefore I could just keep going at that rate longer.
Well, there's no reason to hold your breath unless you're having to dive into the water to do something.
Right. Well, so that's why they teach you how to breathe like one this side. So you're
Right.
really like holding it. You're just breathing it out underwater at the same rate as you can turn your head.
Right.
Or whatever the sport is. But I'm just saying, like, I mean, yeah, sorry.
Okay.
Sorry, but could you move back to the previous slide? I want to ask about the slide 42.
Yep.
Yeah. Can you explain the dorsal respiratory? What is the VRG stand for in that?
This
Yeah.
stands for ventral respiratory group.
Ventral. Okay.
Right here.
Yeah.
That's all that stands for. I couldn't fit it on the slide, so I abbreviated it. That's what that means.
Okay, but can you explain that dorsal? That's
Well, yeah. There's been a lot of debate over the years as to how these two systems work. In looking at various editions of this textbook over the last 30 years, 34 years, every single edition that they come out with, they seem to change their tune here a little bit. So I will just preface this by saying we don't know exactly the whole mechanism to a T, if you will, but we know that there are these major clusters of nerve tissue, one in the pons and two in the medulla. Okay, there's the pontine, this means pons, pontine respiratory group. You can go up here and see it right here in blue. And then below that in the medulla are these two elongated pieces of nerve tissue that make up the dorsal and ventral respiratory group. Aptly named because this is dorsally located, this is ventrally located. Remember, dorsal in the back, ventral in the front. And this is a side view. This is a cutaway view of the brain, right? And the stuff in purple is this. Everybody see what I'm saying? Okay. So it is thought that the pontine respiratory group is communicating via nerve impulses with the medulla, where the ventral and dorsal are. And the ventral seems to take its cues from its close cousin, the dorsal, as well as the pontine group. And collectively, this results in a regulation of both depth of breathing and rate of breathing or rhythm of breathing. This gets complicated, and I don't want to have you really worry too much about intricate interactions between these systems. It gets just too complicated. Just know that there are these clusters of nerve tissue, two in the medulla, one in the pons, that they communicate via nerve impulses with one another to help regulate breathing.
Okay.
Then we talk about partial pressures, and we discussed this the other day. We then get into various factors and how they can influence breathing. We've already talked about PCO2. We've talked about hydrogen ion levels. This describes some other additional factors that can influence how fast you breathe, the rate at which you breathe. All of this impacts particular types of receptors in the body, both chemo and mechano. Chemo meaning sensitive to chemicals. Mechano meaning sensitive to stretch that occurs as a result, let's say, of exercise. So when we talk about PO2, PCO2, a hydrogen ion, we're talking chemoreceptors monitoring those particular levels of those substances. And so now we get into what do the chemoreceptors do? Both central and then there's also peripheral chemoreceptors. And I will say that this section here does require a bit of study and analysis. This is not something you can just read through and know. You've got to really spend some time with it. This is probably the hardest part of the chapter in terms of the physiology. It's not like impossible to understand, but it does require you to spend some time with it. And then we talk about alveolar gas exchange. We've discussed this already, the movement of gases from higher PCO, or from higher partial pressures to lower, whether it's CO2 or oxygen. And then we talk about hemoglobin, what this protein pigment does. This is, remember, found in our red blood cells. There are millions of hemoglobin molecules in each red blood cell. And we can see whether that hemoglobin is charged with oxygen or whether it has more CO2 on board based on the color. Remember us mentioning the other day, Jolene's jacket was bright orange red, and I compared the door color, which was more of a purple, to the deoxygenated, even though we should call this oxygen poor, right? Should really not use that word. These dissociation curves, do not worry about this. This is, again, a very high level physiological concept. I'm not going to take time to talk about it. I think you don't need to know it, worry about it. If you took advanced physiology, you'd have to understand this. I want you to basically have an understanding of how the CO2 is transported in the blood. And we talked about this the other day in review, right? The three mechanisms. So spend some time on that. That's important. What is the chloride shift? And then what happens once we get the blood to the lungs? Well, we reverse those processes so that the CO2 can be exhaled. So quite a bit of stuff in chapter 19. The last chapter was the blood chapter. So let's pull that one up. Do you see the blood chapter? Come on, where is it?
Yeah.
You see the blood chapter?
Yes, I can.
Okay. So this goes through the formed elements. It also describes the other major component of blood, which is plasma. This is the matrix of this connective tissue. Blood is a connective tissue. This is a great slide because it summarizes, you know, the three different kinds of formed elements, and then it subdivides the white cells into gentle Ben and the monocytes and lymphocytes. It also breaks down the plasma a little bit. Even though it's mostly water, there are some other dissolved components here, which I do talk a little bit about in the PowerPoint. Not all of these, but just a couple things here. We talked about fibrinogen and globulins and albumins. We don't talk too much about nutrients and vitamins and hormones, but these, of course, are also housed in the plasma, and that's how these things get distributed throughout the body. We don't talk too much about nitrogen gas, even though it is dissolved in our blood. It plays no physiologic role for us. even though 78% of the air you're breathing right now is nitrogen gas. And then we just basically survey these formed elements, starting with red cells. We describe how their production is regulated by erythropoietin. We talked about this in review, I think. And then we get into more discussion of hemoglobin. Even though we introduced it back in the respiratory system, we are talking in more detail about it in blood, describing the heme group. This is where the oxygen binds to the iron part of the heme group. So on this diagram, you see in the center of these 4 polypeptides that make up the entire hemoglobin, these little kind of flying saucer looking things. Well, where the aliens live is iron. That's where the O2 binds, right at the very center of the double of the flying saucer. So you can have 4 oxygens binding to each hemoglobin molecule. And if you have millions of hemoglobin, do the math. You have 4 times that many oxygen molecules that can be held. So that's why oxygen is such an effective taxi for oxygen. Hemoglobin.
And then hemoglobin can move the carbon dioxide too, right? At a different bonding site.
Right. So I don't know exactly where the CO2 binds. We could find that out in a split second by Googling it. And I don't
want to ask you. That's
yeah. I don't remember.
Okay.
Nor do I remember where the hydrogen ions bind. Remember during the CO2 transport, we talked about how the carbonic acid is split into bicarbonate and hydrogen ions, and the bicarbonate then leaves the blood, goes into the plasma. The hydrogen ions also bind to the hemoglobin somewhere.
Right. Well, so yeah, it was just in the book, it was just saying how efficient that was. And I just wondered, I don't know.
I cannot tell you where the CO2 and the hydrogen ions bond.
No, just that the only reason it was able to was because it was 2 different sites, I guess. It didn't get it.
It's not, well, it's not where the O2 binds. However, I can tell you that carbon monoxide, CO, right, that competes with oxygen for the binding site. That's why when people die from carbon monoxide poisoning, the reason they die from that, which is, by the way, an odorless, colorless gas, you cannot tell. You're in a room with a malfunctioning furnace or whatever, right? You've heard of people dying from CO, right? Poisoning. What happens is the CO binds here where the oxygen would, and therefore the blood can't carry oxygen to the tissues because the CO is blocking it, and the person goes into a coma and dies. It's really interesting, sad, but that's why.
Yeah, it was just one of those. They wrote it really well. I thought it caught my mind.
And then, just to kind of wrap up the class here, we continue in with the leukocytes and then platelets, thrombocytes. And we then get into some more discussion of what plasma is made up of. And then we wrap up the chapter really talking about hemostasis, which is the stoppage of bleeding and the various stages of that, starting with platelet plug formation here, or actually vascular spasm, then platelet plug, and then lastly, if need be, coagulation or clotting of blood if it's a severe enough injury. Sometimes you get paper cuts and so forth where we can stop the bleeding just by vascular spasm or platelet plug formation. We may not need to go beyond that, but if you have a major injury, like a gunshot wound or a bone sticking out of your leg, not to get gross, but then we got to tap into this process, which in and of itself is a rather complex series of events involving various clotting factors that work in a particular sequence that culminates in the formation of a bunch of fibrin threads, like you see here in yellow, that act to trap the red blood cells. And when the red blood cells get stuck up in this thick, sticky network of fibrin threads, that's what stops the blood loss. You don't want this fibrin clot to remain in position forever, and so the body will talk about how at some point in time we need to dissolve that blood clot. Once the tissue's begun to repair itself, then we have to dissolve the blood clot. And there's different mechanisms that we talk about here that help in that dissolving of the blood clot. What you're seeing, by the way, in the news recently is this issue with people having allergic reactions to the Johnson & Johnson vaccine, right? And they're holding it back because of the women that have, one woman has passed away because of it. And what's happening is these individuals are having, they're having these deep vein thromboses, these DVTs. So this is the same issue that relates to the Johnson & Johnson vaccine, where if the blood, now this is a different reason for it. In fact, we don't really know what the COVID vaccine is doing.
Right, because it's only like a very, very small percent.
Yeah. It's less than, I don't know, 0.1%.
Yeah.
But anyway, that's another discussion. What we're talking about here is the fact that if you are sitting down a lot and you have no ability to exercise, what can sometimes happen is the blood can spontaneously clot in major big veins of the body. And there have been people who have gone on, say, over transatlantic flights or fly to Australia or to China. You know, you're in the plane for literally a day. If you don't get up and exercise and you're an older individual, you have a higher risk of these spontaneous clots to form. And then you go and you leave the airport and you go to your hotel and you die of a heart attack because the clot moves up into your heart. Yeah, so next time you go on a deep flight, next time you go on a long flight, get up and walk around. I mean, if nothing else, to avoid getting deep vein thrombosis. Okay. So there was a very, very quick overview of the 4 chapters.
It's pretty awesome though. Thank you.
Sure. Okay. Well, I'll see you guys, I guess, Tuesday and review, right? Have a good weekend. Bye.